go ahead and get started. So just another reminder that we do not have class on Tuesday because it is a university holiday for Veterans Day, so we won't see each other again until next Thursday. There will be homework posted tonight, so as usual, do this, submit it, get the extra credit. There will be discussion sections as usual on Friday and Monday. Some of you may have also noticed that I posted an anonymous feedback survey. It's just a very short questionnaire. And I do this because we still have about half the class left. So if there's some sort of constructive criticism you can give me in a way that I can change the class or change what I do for the better to help you, I want to hear that now so that it can actually be useful to you. So, you always get the evaluations at the end of the class, and that's great for the next year's class. But right now, if there's something I can do, please let me know. Any questions? All right, so let's review what we talked about in the last class. So we began by talking about the other transcriptional systems that we need to make the RNAs we're going to need for translation. And so first we talked about this long ribosomal RNA precursor, and which is the polymerase that makes this one type of RNA? RNA Polymerase I, exactly. And its job is just to pump out tons of this pre-ribosomal RNA. And so that's how it is synthesized. And then we saw there's a number of processing steps. And so in the end, you get 3 mature ribosomal RNAs cleaved out of this. It undergoes modifications. There are methylations that happen at specific positions. There's this weird U turning into pseudouridylation. And what did we call the molecules that guide the enzymes to the ribosomal RNAs to do these modifications? Think about where these modifications happen, where ribosome biogenesis happens. It's in the nucleolus, right? And so these were called snRNAs for small nucleolar RNAs, and they serve as the guides to tell the machinery where to put those modifications. We then talked about tRNAs, so these other RNAs that we're going to need to bring the amino acids in for translation to occur. Which polymerase makes tRNAs? 3, exactly. So Pol I makes this one long ribosomal RNA. Pol II makes the messenger RNAs. Pol III makes tRNAs. And then there's a number of different processing events that need to happen to take the pre-tRNA into the mature form that can actually function in translation. There's this RNase P enzyme that trims off the 5' end of the tRNA. And RNase P was interesting because it contains 2 subunits, a protein and an RNA. And it's actually the RNA that does the enzymatic activity of removing this 5' leader. What do we call RNAs that have catalytic activity? Ribozymes, right? Sounds like an enzyme, but it's made of ribonucleic acid, so ribozymes. We then also get trimming of the 3' end, and then this enzyme that puts specifically CCA onto the 3' end, because that's where the amino acid is going to get attached. Some tRNAs, but not all, have an intervening sequence or a little intron. This gets cleaved out by an enzyme complex that is entirely different from the spliceosome that takes introns out of our messenger RNAs. And finally, there's a number of different base modifications that happen and are necessary to make a mature tRNA. Let's do a clicker question. Which of the following statements about eukaryotic translation are correct? 10 more seconds. All right, all answers in. Oh boy. All right. Is it more than one of the above? Let's go through and see if that is the answer. Who wants to share their thinking about this question? Khalil.
I said A because I remember the last part.
So you said E, so you like more than one of the above. Which one was your favorite?
E was correct according to what we studied
All right.
at the end of the class.
So let's look at A. So A says, counting from the 5' end, the first base of the codon and the third base of the anticodon are the wobble positions. Is that right? It's the opposite. Yes. Yes. Is that what you're going to say, Isabel? Yes, exactly. So remember when we talked about the wobble position? So wobble means you're allowed to have non-standard base pairing at that position. And if I ask you to tell me the base pairing, I would give you one of those little charts to tell you what non-standard is allowed. But what you need to remember is which bases are allowed to wobble. So it is the first position from the 5' end for the tRNA anticodon. If you can just remember that part, the other part is automatic, right? Because the first position in the anticodon has to be base pairing to the third position of the codon. So those are the only pairing that is allowed to wobble. The other parts of the codon-anticodon base pairing, they don't wobble. Just use your normal base pairing rules for that. So A is not correct. Everyone's clear about that? Right, so it was reversed, exactly. B says the first 5' AUG, 3' usually sets the reading frame. Correct? All right, we don't want to use the word always in biology. That's why I put usually. The way that we taught in this class, and every example I'll ever give you, will start with the first AUG. But in reality, there are differences to that. Sometimes the first one is skipped because there can be regulation. But the way I've written this, the first AUG usually sets the reading frame. Yes. So that one is correct. Pretty standard the way we learned it. What about C? So C says the ADS ribosome along with the initiator, methionine tRNA, hooked onto methionine, and eukaryotic, that's what that little E stands for, initiation factors bind the mRNA 5' cap and scan for the start codon. Is that correct? Isabel? Exactly. So remember, in pre-initiation, the two subunits of the ribosome are not together in the beginning. And it is the small ribosomal subunit, 40S in eukaryotes, that comes with the initiation factors and the initiator tRNA hooked onto methionine. That scans until it finds, usually, the first start codon, that AUG. It will pause, and only then does the 60S join. And when we have 40 and 60S together, that is then the ADS ribosome. So don't add up 40 and 60. It's based on sedimentation. And so this would be correct if I had said the 40S subunit. But it says ADS. And so I'm not changing these little things to try and be tricky. What I'm trying to do is to get you to pay attention to every word in the question and think about, does that make sense? Is that correct? So does everyone understand why C is not correct? All right, let's look at D. D says translation terminates when release factor tRNAs recognize a stop codon. Is that true? Hearing yes. Everyone likes that one. Some of you only voted for B. So some of you didn't like this. Read every word. Yes? Yes, exactly. There are no tRNAs that will base pair and have an anticodon for the stop codon. Remember, there is this release factor 1, which is a protein that is shaped like a tRNA. It is made of amino acids, but it's able to fit into that A site and say, we got to stop translation. So if this had said release factor proteins, it would have been correct. But it is not tRNAs. Does everyone remember that kind of now? Make sense? All right, so the only correct one is this first AUG usually sets the reading frame. Any questions about that? All right, so we are going to review the steps of translation, and so this will be familiar to you all. So when we talked about translation, what we said is that the codons, which are the nucleotides, read in groups of 3. And the way that they are read is that the anticodon will base pair to the codon. The tRNA is attached to a specific amino acid on its 3' end, and that is how the correct tRNA, or the correct amino acid, is actually recruited. What did we call the enzymes that will bind a specific amino acid and then all the tRNAs to which it is supposed to be attached? Anyone remember what we called it? Yes? Synthetases, yes. So aminoacyl, it's putting an amino group, it's acylating the tRNA. So it's sort of synthesizing the charged tRNA or the tRNA with the amino acid. So there are 20 different synthetases for the 20 different amino acids. So let's look at these steps of translation. So as we talked about in the clicker question, we start with initiation. We have to assemble the pre-initiation complex. There are initiation factors that recognize that 5' MC7G cap. Their assembly there is often aided by poly-A binding proteins that loop the RNA around and help stabilize interactions of initiation factors with this 5' cap. Also part of the complex is the small ribosomal subunit, which would be 40S in eukaryotic cells. And then with that ribosomal subunit, you will have the initiator tRNA that is attached to methionine. This complex will then scan through the 5' UTR until it reaches usually that first start codon, that first AUG. And so the complex moves along. As soon as it reaches that first AUG, the complex will pause, and it pauses so that 60S can then come and join, and you have now a full ribosome, an 80S ribosome that is capable of doing translation. Remember, the initiator tRNA was in this P site, so that means we have an empty A site for that first step of elongation to occur. That will occur as tRNAs are sort of sampling that codon to see if it can have stable base pairing to the anticodon. Once there is stable or correct base pairing, there's a conformational change that sort of hooks the tRNA into that position, and then you're able to do the enzymology of translation. What is the actual enzymology? What is the reaction that is happening on the ribosome? What is the whole point of having translation? It's to make protein. What's protein made out of? Amino acids. What do you call when you link two amino acids together? It's a peptide bond, right? So you're going to have peptide bond formation. So let's look at this peptide bond formation that is happening on the ribosome. So when you initially have your initiator tRNA methionine, so the superscript here means this is the tRNA that encodes for methionine. When there is a dash and the code for an amino acid before it, that means that this tRNA is still attached to an amino acid. So this is attached to methionine right here. The next position encodes for a serine. So there is a tRNA that encodes for a serine that is still attached to its serine amino acid. And then here is where the reaction is going to take place. So this amine group on the attached serine is going to attack this ester bond right here, And that is what's going to allow then for peptide bond formation to take place. When that peptide bond formation is made, look what happens. It severs the amino acid from that 3' end of the tRNA. So you now have what we call an empty or a tRNA that no longer is attached to its amino acid. And temporarily in this A site where the peptide bond has happened, we now have a tRNA that is attached to its original serine and the peptide bond it has formed to the methionine here. Do you guys see now why we call the beginning of a protein the N-terminus or the amino terminus? Here's the amino terminus, right? We're always going to have this free amine group. You see why we're going to call the end of a protein the C-terminus or carboxyl group? At the very end of translation, this is going to get severed from the tRNA, and you're always going to have this free carboxyl group. So that's what we mean by a protein having an N-terminus and a C-terminus. All right, so this reaction is going to continue. So you're going to have translocation because this empty tRNA, we want to put it in the exit site so we can get rid of it. The growing peptide is now going to be in the P site. We now have an empty A site, so the next tRNA can come in, base pair its anticodon to the codon, and then you can do that next peptide bond formation reaction. And then that will continue until there's a stop codon in the A site. Once there is a stop codon, this is recognized by these release factors. RF1 is a protein that recognizes any of the 3 different stop codons. It will come into this A site. It has a helper, this RF3 protein, which is attached to GTP. When this complex comes into the A site, that then allows for translation termination to end. And so there's several things that need to happen. You need to release the peptide bond, so you need to sever it from that tRNA where it was in the P site. That protein gets released. And then you also need to disassemble the ADS ribosome. There's a set of factors, ABCE1 is one of them, that helps blow apart this 40 and 60S so that they can be recycled. The tRNA is released, can go back to a synthetase, get recharged with another with its correct amino acid to allow the process to start over again. We said that often messenger RNAs can have multiple translating ribosomes on them. So as soon as the first complex clears that AUG, you can have another pre-initiation complex come on and then another one. What do we call that in RNA associated with multiple ribosomes? Anyone remember? Multi, many, poly. Polysome, right. The full word polyribosome also means the same. We often shorten it to polysome. So that's when we have multiple ribosomes riding on that messenger RNA. And what that means is one messenger RNA can give rise to many proteins. Any questions about translation? All right, so we have now learned our 3 major molecular mechanisms. We know how DNA gets copied into more DNA through the process of replication. We know how DNA can get copied into RNA through the process of transcription. And finally, how messenger RNAs are decoded into protein through translation. But of course, there's a lot of regulation that goes on to determine, well, once you have that messenger RNA, how stable is it? How well is it going to be recognized by the ribosome? How much protein are we actually going to be able to make? And that is the topic of tonight's lecture. And so this is all called post-transcriptional control of gene expression. So what do we mean by post-transcriptional control? So what that refers to is what happens to the RNA after it's been transcribed. And in fact, all of the examples, well, most of the examples we'll talk about tonight is it's been transcribed, it's been spliced, capped, polyadenylated, it's gone out to the cytoplasm, and then how is it going to be regulated in order to make proteins at a certain level, as well as of a particular sequence. And so first we'll talk about modifications that can happen to messenger RNA. We sometimes call this editing. This changes the base in a way that either it will be read as a different nucleotide or there will be RNA binding proteins that will specifically recognize the modified version and regulate the RNA that way. We'll talk about basic factors that regulate how stable an RNA is. There are processes that can recognize if an RNA is aberrant, if splicing has gone wrong, if it hasn't gotten a cap. We call these surveillance mechanisms to get rid of these RNAs that can cause problems. Then we'll talk about some mechanisms of translational control. So once you have the RNA, how well is it going to be translated? There are situations where essentially all the RNAs in the cell will be upregulated for translation or downregulated. That's what we mean by global, essentially everything in the cell. And then we'll talk about some specific situations where only RNAs that have particular sequences or elements that are bound by specific proteins undergo this type of regulation. And then finally, we'll talk about this fact that some mRNAs can be localized to different parts of a cell to allow for local translation or local production of a protein where it is most needed in the cell. All right, so what do we mean by RNA editing? So this would be after transcription. So the polymerase has copied the template, you have your RNA, but then you can change the sequence of an mRNA through specific enzymes that are able to do this. The change could be a deletion, it could be insertion, or actually changing the identity of the base itself. And these editing enzymes will usually recognize a very specific sequence in the RNA or a structure. Sometimes the RNA, remember, can fold back on itself. And so some of these editing enzymes will recognize the structure, sometimes sequence and structure, to know this exact nucleotide is what we want to change. So here's an example. This is your ApoB gene. And so the ApoB gene is pretty long and complicated, so all these blue boxes are the exons. You make two different versions depending on which tissue is making it. So your liver cells produce the long version of ApoB protein, this ApoB 100. And this is important because this version of the ApoB protein takes cholesterol from your liver and transports it throughout the body. In your intestines, you have a shorter version of this ApoB protein, ApoB 48. And this version of the protein doesn't pick up cholesterol. Instead, it picks up triglycerides from your intestine and transports those around your body. So, two different versions of the protein that was made from the same exact gene and initially the same messenger RNA. What is different? How do we get these two different size proteins? Well, it turns out that in your intestine, there is an enzyme complex called ApoB-EC, which stands for ApoB editing complex. This ApoBec complex recognizes a specific position in the messenger RNA, and exactly at position 20, actually this is the protein position, but exactly at this position here in exon 26, it converts this C to a U. CAA encodes for glutamine, an amino acid. What happens when you change that C to an A? You now have UAA. What is that? It is a stop codon. So when this messenger RNA is recognized by the translation machinery in the intestinal cells, it'll start translating. It'll see this stop codon here, and it'll stop translating. That's how you make a shorter version of this protein in your intestine versus in your liver cells. It is an example of editing, changing the sequence of the nucleotide. Here's another example of editing that's much more prevalent. So what base is this? Yeah, an A, adenosine, exactly. So adenosine can actually go through a deamination reaction. It is catalyzed by an enzyme to produce this base. Anyone know what that base is? It's actually inosine. So we mentioned this when we talked a little bit last time about inosine being in tRNAs. So this is in messenger RNAs, and there is an enzyme that does this editing event. It is an adenosine deaminase acting on RNA. That's what ADAR stands for. And so these ADAR enzymes will recognize adenosines at very specific positions in messenger RNAs and catalyze these deamination reactions. When you have an inosine in messenger RNA, it's read as a G. So you're going from being read as an A to now being read as a G, which could change, it will change the sequence of the codon and could potentially also then change the amino acid that would get incorporated at that position. Here's an example of that. So your glutamate receptor messenger RNA is a substrate for ADAR activity. Your glutamate receptor is very important. It's a calcium channel and needed for neurotransmission. And you need different versions of this channel at different times in development. And one of the ways you get different versions is through an editing event. So there is a site in the glutamate receptor messenger RNA. We call it the RG editing site. It changes this AGA to an IgA. Given the name of this edit site, anyone want to guess what AGA encodes for? What amino acid? So an amino acid, R, what is R? Arginine, right? So, arginine, this big charged amino acid, gets changed to an I. That I is read as a G, and then it encodes for G. What is G? Glycine. You couldn't have two more different amino acids, right? So this really changes the property of this calcium channel, and you need both versions at different times. It's not a mutation. It's not a bad thing. You actually need editing to have these different versions of your glutamate receptor. And in fact, patients who have defects in A to I editing often suffer schizophrenia, seizures, and other problems, some of which are probably linked to problems with their balance of calcium channel receptors. All right, so here's an interesting thing. So I'm showing you the actual sequence that is recognized by ADAR to do this editing event. And so I can see, looking here, I see an A here, I see a G here, then I see a G. Wait a second, I just told you it's AGA to IgA. Did I make a typo at this third position? Where is this A coming from? Yes? Branching out over here. It's probably coming from somewhere else. So I'm telling you this is a 5' splice site. Where is that third A coming from? So before this gets translated, what's going to happen to this messenger RNA? So you edit it, and then what? You're going to splice it, right? So if I'm telling you this is a 5' splice site, this grayed out area, this is the intron. This is the intron, so that gets removed. So what do you think the nucleotide is, the first nucleotide of exon 14 down here? It is an A, exactly. That is where the A is coming from. So there's a few important things to take away from this. First of all, the reason why I included the intron here is because that's what this ADAR enzyme actually recognizes. It is recognizing the pre-messenger RNA that has this sequence and structure. So it's important to have this A here, but it still needs to be attached to the intron. Otherwise, the enzyme won't recognize it. The other thing to realize is after you have spliced in the nucleus, you don't have your codons all together in the same exon necessarily, right? You can have the first two nucleotides of a codon here, and then the third nucleotide in the exon downstream. Because the reading frame is not set until you have your mature messenger RNA that goes out to the cytoplasm, and only then does translation start. Question? Some editing events happen to mature RNAs. Some happen when they are still pre-messenger RNAs, before they have even undergone splicing. So it's Depends on the RNA and the actual editing event. This example is definitely on the pre-messenger RNA. Any other questions about this? All right, so here's another type of editing event that we really just learned about over the past 10 or 15 years, and it turns out to be quite common. So again, which base is this? Adenosine. We're still looking at adenosine, but now instead of converting it to inosine, what has happened to it? It is methylated. So this is called M6A because it is at the 6th position that you have added this methyl group. So methylation on the 6th position of adenosine. And there are a variety of enzymes that are capable of catalyzing this type of editing. And so in general, we call the enzymes that put the methyl group on M6A writers. So they are writing the modification. They are putting the modification on the RNA. It actually can be a reversible modification. So we also have enzymes that we call M6A erasers. So we'll remove that methyl group to bring it back to a normal adenosine. And this still is read as an adenosine, but the whole point of the modification is there are RNA binding proteins that we call M6A readers that will only bind to the RNA when it has the methyl group. And it's these readers that are going to determine the fate of the RNA that has that modification or not. And so that brings us to this question. So if there's all this effort going into putting this methyl group on, taking this methyl group off, what is the effect on the messenger RNA? And the answer actually boils down to it really depends on the reader. So there are different readers in the cell that will read the M6A and also often nucleotides flanking that M6A, and that's how you get some specificity. And sometimes these readers are in the nucleus, and they can regulate things like splicing and nuclear export of the RNA, and sometimes enhance splicing, sometimes reduce splicing, sometimes promote export, sometimes reduce export. There are other readers that are in the cytoplasm that can recognize RNAs that have this modification. They can definitely affect the stability of the RNA, and it's still a little controversial whether they are directly regulating translation of the RNA or not. So in these cases, it's really specific to the particular RNA, the modification, and the sequences around it, and which reader is actually available in the cell to bind to that modified version of the RNA and determine is splicing going to happen here or not, or is the RNA going to be stable. And that brings us to the topic of RNA stability. And so whenever we're talking about the concentration of a messenger RNA in the cell, that's really going to be determined by synthesis. So how efficiently is it transcribed? How much is the cell making? Balanced by its destruction, so degradation of the messenger RNA. Once you make a messenger RNA, it doesn't live forever. It gets degraded eventually. And there are different rates of degradation of the messenger RNA. And that's what we mean by mRNA stability. So in prokaryotes like bacteria, mRNA half-life is often very short, and that's because the replication cycle of a bacteria cell is very short. In eukaryotic cells, like our own human cells, it can vary dramatically. Some also are only minutes. Others can last for hours. Other messenger RNAs we think last for days, in particular human cells. And so there's going to be a lot of things that are determining how long that RNA sticks around. One element that we know typically promotes degradation or destabilization of a messenger RNA are what we call these AU-rich instability elements. And so here's an example, as the name suggests. It would be a little sequence of RNA that has a lot of A's and U's together. It doesn't always have to be A, U, U, A. It could be U, A, U, U, A, but a region that is rich in AU sequences. They are typically in the 3' untranslated region. That's typically where they're going to be recognized. You often will have multiple copies in that region. So the odds of them recruiting the destabilizing proteins are going to increase. So more AU-rich instability elements, probably the less stable the messenger RNA is going to be. And what it does is it recruits proteins that very often recruit enzymes that are going to chew off the poly A tail of those messenger RNAs. And so that then brings us to the three very basic mechanisms of how you would generally degrade a messenger RNA. So in the first model, we have what's called deadenylation-dependent messenger RNA. We think this is the most common general method that's used in our human cells. So remember, a mature messenger RNA is going to have this funny M7G structure on its 5' end. It's going to have a poly A tail on its 3' end. And there are proteins that bind to those modifications to stabilize the RNA. But they're not glued there, right? There's some motion. There are things that can happen so that it's not always protecting those sequences. And in fact, there are deadenylase enzymes that start to get in, knock off those poly A binding proteins, and chew up the poly A tail. We generally call these deadenylases. So once that enzyme gets on there, gets rid of the poly A tail, then it's just kind of like a Pac-Man. The way it's drawn here, just chomp, 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 chomp. It's just going to start chewing up from the 3' end. So we call this 3' to 5' decay because you're moving from the 3' end of the RNA towards the 5' end. The other thing that happens when you remove that poly A binding protein, remember the poly A binding protein can also interact with factors that recognize the M7G cap of the messenger RNA and that sort of loop structure. So if you lose those poly A binding proteins, you don't have that interaction anymore, which makes the cap more vulnerable for degradation. There are enzymes called decapping enzymes that can then come in, chop off that 5' M7G cap. Then there are other enzymes, other nucleases that are then going to degrade the mRNA from this direction. So this is going to be from the 5' towards the 3'. So we call it 5' to 3' decay. So basically you're getting your RNA chewed up from both ends. It's going to be unstable. Once you get rid of the cap and the poly A tail, you get very rapid degradation of that mRNA. Now, there are some situations where you don't even need to deadenylate the RNA. There can be sequences in the RNA that recruit particular RNA binding proteins that then can directly recruit this decapping enzyme. Again, once you chop off that 5' end, you bring in nucleases that are going to rapidly degrade that messenger RNA in the 5' to 3' direction. If we can degrade our RNAs starting from the 3' end, starting from the 5' end, there's only one other option, and that's if we cut our RNA in the middle. If there is a nuclease, so ones that cut RNAs in the middle, we call endonucleases. Those that work from the outside, so from the 5' or 3' ends, we call exonucleases, so chew in. Endo cuts in the middle. If you cut your RNA in half, well, you don't have any protective structures there, right? And so immediately you can get nucleases that are going to chew up the RNA in this direction. It can still have its cap here. It doesn't matter. You're going to chew up the RNA or chew it up in this direction. It doesn't matter if you have your poly A binding proteins there. You've now made the RNA very vulnerable by cutting it in half. No protective structures there. You can degrade the messenger RNA. Any questions about that? Yes, Antonio.
So these degrading proteins, are they just general or do they depend on modification?
They are largely general. So eventually an RNA is going to be degraded in the cell, no matter how stable or not. And these are sort of the general pathways. But you can have RNA binding proteins that are really good at recruiting a deadenylase or recruiting the decapping enzyme to the messenger RNA. And then you're going to get regulated or rapid degradation. And we'll see a couple of examples of that in upcoming slides. Anything else? Yes? Yeah, so this particular DCP1 and 2 specifically chops off that weird bridge, that triphosphate bridge. And what it leaves behind is a 5' monophosphate. which then this exonuclease called XRN is able to just rapidly degrade from. All right, so now let's move on and talk about RNA surveillance. So these are sort of quality control mechanisms that our cells use to recognize when an RNA hasn't been properly processed or there's something wrong with it, and you want to degrade it before it might be translated or cause other types of problems in the cell. So in the nucleus already, if there's improper processing or splicing, those RNAs usually are retained in the nucleus and degraded right there. And there are different ways that your cell can recognize, whoa, the spliceosome's sitting too long on this messenger RNA. Something must have gone wrong. Let's just degrade that messenger RNA. The other type of surveillance we have is actually when we have a situation where an RNA has what's called a premature stop codon. So we know what our stop codons are, right? All of our messenger RNAs will have a stop codon where we want translation to end. Another word for a stop codon is a nonsense codon. So when you hear this word nonsense, that just simply means a stop codon. RNAs that gain a premature stop codon are usually targeted for efficient decay. So what do we mean by a premature stop codon? So a normal messenger RNA will have a stop codon at the end of the reading frame. Anytime you happen to have a nucleotide change that puts a stop codon earlier in that messenger RNA, it will potentially be recognized as aberrant and undergo decay. So how could you get this? Well, if you had a mutation in the DNA and it encoded instead of a CAG, a UAG at this position, there would be a stop codon. If transcription makes a mistake and puts in the wrong nucleotide, you could get a stop codon. Or if we have changes in splicing, an incorrect splicing event, sometimes that can put a stop codon into frame. So let's think about why it's so important to get rid of these types of RNAs that might have a premature stop codon. So they look otherwise like a normal messenger RNA and would go out to the cytoplasm and be translated. But the end result, of course, would be you would get a truncated protein. This could be very dangerous for certain types of proteins, right? If you make a truncated version, it could take on a dominant function. It doesn't have all of its protein and therefore might not be regulated the same way. Here's sort of an example that goes back to what we talked about right before the midterm. So what if we have a particular type of transcription factor that has its DNA binding domain, a spacer domain, a ligand or hormone binding domain? What is this encoding for? What kind of protein? What kind of protein did we talk about? That's DNA binding domain and a ligand or hormone binding domain? Nuclear hormone receptor, right. So remember, these were these transcription factors that have a domain that is going to bind to DNA, and it's very important to be able to bind to the hormone in order to regulate and actually upregulate transcription. If you are missing the regulatory domain, the hormone binding domain, you could have this truncated version of this transcription factor, DNA binding, bound to its DNA binding element, and never be able to activate. This could cause a lot of problems for transcription. And so you can imagine many different types of proteins could be dangerous if they are made in the truncated version. So we have a mechanism for recognizing these RNAs that have premature stop or premature nonsense codons. It's called nonsense-mediated decay, or NMD for short. But first we have to consider this problem. So how does a cell actually know that there is a premature stop codon, right? The sequence of the premature one might look exactly like, it might be the exact sequence as the natural one. So how can a cell actually detect that there is this aberrant or earlier premature stop codon? So we're still trying to figure out the answer to this, but there is one model that explains a lot of nonsense-mediated decay, and that is based on what we call the exon junction model. All right, so what does this mean? It turns out that when your pre-messenger RNA is undergoing splicing by the spliceosome, every time an intron is removed and the exons are ligated together, there is a complex of proteins called the exon junction complex. It's multiple proteins that gets deposited right next to where the two exons got ligated together. So after an RNA has been fully spliced, it will have multiple exon junction complexes that have marked everywhere exons have been ligated. This messenger RNA, along with its bound exon junction complex proteins, goes out to the cytoplasm. In the cytoplasm, it'll be recognized for the first round of translation. What happens in that first round of translation? Well, we get the ribosome coming on, starting to translate this messenger RNA, and the ribosome's big and powerful, and it's going to knock off this exon junction complex as it's reading the open reading frame until it comes to a stop codon. When the ribosome comes to a stop codon, the proteins, the release factor 1 is going to come into that stop codon, cause the ribosome to disengage, and you're going to stop translating at that position. What does that mean? That means any downstream EJCs that are downstream of that stop codon are still going to be stuck to the messenger RNA. They were not removed by the ribosome because the ribosome stopped at that first stop codon. Why is that important? Turns out that these EJC proteins recruit deadenylases to messenger RNAs. So if you keep EJCs on your messenger RNA, they are going to grab these deadenylases, they are going to chew up that poly A tail, 5' cap is going to become vulnerable, and you're going to get very rapid degradation of that messenger RNA. So yes, in that very first round, or what is sometimes called the pioneer round of translation, you might make one truncated protein, but because of this mechanism, because the mRNA is then going to be degraded, you're not going to make hundreds of that truncated protein. Does this make sense to you, this exon junction model? Here's a question. What if my premature stop codon is here in this last exon? Is this model going to work? No. It still is premature. It is before the natural one, but this mechanism will not work because this EJC would have gotten removed before that stop codon were recognized. So if premature stop codons end up near the natural stop codon, usually this mechanism doesn't happen, but usually you don't end up with a very truncated protein. So it's not as dangerous as having premature stop codons closer to the beginning of the messenger RNA. Question?
I was going to ask what the stop codon was like if the red one, and we still end up with a protein. Will that protein still be functioning protein?
It depends all on the messenger RNA. Sometimes when you make a truncated protein, it can't fold correctly, and it will just get degraded anyway. But sometimes you can make a truncated protein, and they can be very dangerous for the cell. All right, here's another question for you. What if we have splicing happening? So we have an intron in the 3' UTR. Can we have an intron in the 3' UTR? So what does 3' UTR mean? Untranslated region. Can you have an intron in there? Why not? An intron is just a sequence that needs to get removed. You could have one in a 3' UTR. You could have one in a 5' UTR, right? Does that make sense? So it's just part of the messenger RNA. So now let's say, so all this sequence from here to here would be 3' UTR, right? Let's say we had a little intron in here. It got spliced out in the nucleus. What would be bound here? An EJC, right? So the EJCs only know splicing happened here, exons got ligated together, I'm going to bind. You stop translation here, what is that EJC going to do? It's going to stay, exactly. There's no ribosome that's going to translate through a 3' untranslated region. RNAs that have introns in their 3' UTR are very unstable because the exon junction complex can recruit the deadenylase enzyme. And for some RNAs, that is a natural state. It is important for them to be lowly expressed, and that is one of the ways they keep their levels low. Question, Antonio?
What if some of the RNAs, like if they're all trying to do splice and they want to early stop codon, what if they can guarantee it to be created?
Yeah, so some of you might even be thinking back to, wait a second, that ApoBec example you talked about. That was definitely causing an early stop codon. There are a lot of exons after that. The way that works is, along with, it does have EJCs and all of that, but there is a protein complex that comes along with the editing enzyme that binds to that RNA and actually protects it so that it can be translated. So there are ways to overcome this if the cell has evolved to make the mRNA in this isoform this way. Other questions? All right, so now let's move on to talking about some examples of translational control. And so what we're talking about here is we have the messenger RNA, and how well is it actually going to be recognized by the ribosomes to pump out protein? The first example is what we call global translational control. And when we're talking global, that simply means essentially all the messenger RNAs in the cell are either upregulated for translation or downregulated. So you're going to have higher levels of translation or lower levels of translation. The way that translation is often globally regulated, so if you want to globally turn down translation, this is often at the level of initiation. So don't even bother starting to scan and look. We're going to block things at the initiation level. One way that your cell is able to couple growth, so growth is going to require more translation, high levels of translation. And so you always want to couple that to situations where there are signals and nutrients available for the cell to have high levels of translation. So there is a kinase called TOR that regulates translation in response to nutritional status. And don't worry about all the various proteins and the way that this pathway is regulated. Simply remember that this kinase, when it's active, that is when we have increased protein synthesis. So it affects a number of different pathways, so you get higher levels of translation. Some of the downstream things it does is it pumps up ribosome biogenesis. So you want to do more translation? Let's make more ribosomes. If we have more translation, we're going to need more tRNAs. So let's increase Pol III transcription so we get more tRNA. So active TOR means we have higher levels of translation. And in fact, we talked before, cancer cells often want to have high levels of translation. Cancer cells often activate TOR, activate these pathways, and therefore this is often a target that we think if we can turn down TOR activity specifically in cancer cells, maybe we can block them from rapidly reproducing. So this is just a general idea of how global translational control can occur. Now let's look at a specific example. And so this specific example involves something interesting. It is cytoplasmic polyadenylation. Where does polyadenylation normally occur? In the nucleus, right? So it can even be co-transcriptional, right? Those factors ride along with the CTD of the polymerase and polyadenylate the RNA. The RNAs go out to the cytoplasm. Well, in certain cell types, there can be this mechanism of regulating translation by regulating polyadenylation in the cytoplasm of messenger RNAs. So it turns out that eggs, like human eggs that are going to be ready for fertilization, they are not transcriptionally active. All the RNA they have in them came from the maternal stores, came from the oocyte. And they are packaged in that egg in a sort of translationally dormant state. They're not being translated because it's not time yet, but the RNA is there because we're going to need it very rapidly once fertilization occurs. The way that they are kept in this translationally dormant state is that they have a short poly-A tail, sometimes only about 20 nucleotides of A's on them, and they will have an element or a sequence in their 3' UTR called a CPE. CPE stands for cytoplasmic polyadenylation element. The CPE is recognized by an RNA binding protein called CPEB, cytoplasmic polyadenylation element binding protein. This protein will bind all those maternal RNAs that have this sequence in them, and this protein binds to a protein called maskin. What maskin does is it interacts with some of the initiation factors that are needed for translation. And in doing this, it blocks the normal initiation factors from binding to the 5' end of the messenger RNA. So remember when we talked before about one of the mechanisms for efficient translation is that the 3' poly A tail has these poly A binding proteins that then interact with initiation factors, and this helps promote initiation of translation. Well, when you have maskin there and you don't have a poly A binding protein because this tail is so short, you can't get the correct initiation factors recognizing the 5' end, and so you're just sort of trapped in this dormant state. No translation is going on. You're just holding on, stabilizing that messenger RNA until when it might be needed to pump out protein. So how do we change this dormant state? Well, in the case of the egg, when there is fertilization, fertilization causes hormonal changes. One of those hormonal changes causes kinase activity. And this kinase, one of its targets, is this RNA binding protein. So this CEPEB gets phosphorylated. What does that do? Well, it has two major consequences. One is it is now able to recruit cytoplasmic polyadenylation machinery. So there's a cytoplasmic polyadenylation polymerase in these eggs that are then able to add a long poly A tail to these RNAs. Then poly A binding protein is able to bind to those tails. The other thing that phosphorylation does is it causes release of maskin. So maskin was masking that 5' end of the RNA, not allowing it to interact with the correct initiation factors. You kick out maskin, you now have poly A binding protein here, and you can now recruit the normal translation pre-initiation complex. And so what this allows for is very rapid protein synthesis. You already have the RNA there. It's already been spliced and modified. It's just sitting there in the cytoplasm waiting for this signal to then allow for translation of these specific messenger RNAs. This very similar mechanism actually also occurs in our neurons for certain messenger RNAs, and it's very important for translational control of learning and memory. So as you probably know, your neurons, you have the cell body, the nucleus, but there's these very long projections or dendrites or axons that can be quite distant from where RNA synthesis occurs. And so what can happen is you can have messenger RNAs stored at the very ends at these synapse positions so that when there is a signal, the RNA is already there and you can very rapidly translate it. And the way that this works for some messenger RNAs is that they contain CPEs in their 3' UTRs. The neurons have this cytoplasmic polyadenylation binding protein that is bound to the CPE. It will keep those RNAs dormant in sort of a masked state. There's a different name of protein that actually works in the neurons, but it works like maskin until there is a signal. So the signal can be from the presynaptic cell saying, hey, we need to make this protein so that you can remember something in molecular biology. And what will happen then is that the signal will cause a kinase to phosphorylate CPEB. It kicks off that negative regulatory protein. You get polyadenylation, and now you get efficient translation. And so this is really cool, right? Because otherwise, if you didn't have this local translation and the signal came into the cell, it'd have to go into the nucleus. You'd have to unpack that chromatin. You'd have to transcribe the messenger RNA. You'd have to do splicing, modifications, get the RNA out to the cytoplasm, get it maybe make the protein in the cytoplasm, and then get the protein all the way out to where it is needed. That would be a very slow process compared to this localized translation. Any questions about that? All right, so now let's look at another example of how one specific RNA binding protein can regulate the translation and stability of two different types of messenger RNAs, of two different genes that are essential for iron homeostasis. So iron, very important for our bodies, right? Why do your cells need iron? Hemoglobin, yes, very important for that. And not even, yes? Cofactor for what? It's actually a cofactor for many enzymes, but electron transport. So all your mitochondria really need this. You don't want to be low in iron, right? You're iron deficient, you have low energy. It's because of your red blood cells, but also electron transport. So you want to make sure you don't have too low of iron. And so the way that this is regulated, if you need to increase the amount of free iron for your cells to use, you have a gene that encodes a transferrin receptor. This is in the membrane of your cells. It brings iron into your cells. So the receptor brings iron into the cells. There's another protein called ferritin. Ferritin is like an iron sponge. It soaks up all free iron. So if you're low on iron, you don't want to have very much ferritin around. It's going to be sopping it up, and you're not going to have it available. So in low iron conditions, you want your receptor on. You want this sponge ferritin to be off. On the other hand, you don't want to have too high of iron because it can lead to there can be toxic levels of free iron. What does too much iron do to your cells? Yeah, oxidative stress. It splits oxygen. You get free radicals. Free radicals go around, poke holes in your DNA, your membranes, everything. That's a very bad situation too. So you want to control high iron. If you already have high iron, you want to turn that receptor off. You want to turn the sponge on so you can soak up that free iron. So how are these two different genes that sort of have opposing functions regulated? They are regulated, they are regulated by an RNA binding protein called the iron response element binding protein, or IREBP. This RNA binding protein, when it is bound to iron itself, so this RBP, this RNA binding protein, it can bind to iron. When it is bound to iron, it cannot bind to RNA. So that's key to remember here. So when there's a lot of iron around, it's going to be bound to this RNA binding protein. That protein can no longer bind to RNA. The other thing we'll see is the location. So IRE, these are these sequences. So iron response element are these sequences that are going to be in the ferritin RNA and the transferrin receptor RNA to which this RNA binding protein will recognize and bind. And so we'll see that the location of these can also be important for determining the mechanism of regulation. All right, so we're going to look at ferritin first. So remember, ferritin is this sponge that's going to soak up high iron. So if we have high iron levels, we want to make a lot of ferritin, right? So for ferritin, its IREs are in the 5' UTR, right? That's easy to remember. Ferritin begins with an F, 5' UTR, F. So ferritin, 5' UTR, has these IREs. And the reason that they're drawn as sort of these lollipops is because they are these stem loop structures. So they fold back on themselves a little bit. And that's important for the protein to be able to recognize these sequences. So remember, when we have high levels of iron, it is going to be bound to that RNA binding protein. Can't bind to the RNA. So IREBP is out of the equation. We have the ferritin RNA. The ribosome is going to come through, plow through these little stem loops. It's going to decode the messenger RNA and make lots of ferritin. All right, so you're making lots of ferritin, you're bringing free iron levels low. Now you're going to have a situation where IREBP, you're going to have some molecules that no longer will be bound to iron. When this RNA binding protein isn't bound to iron, it can now bind to RNA. It's going to bind to those sequences in the 5' UTR of the ferritin messenger RNA and form very stable complexes. What does that mean? That means when the pre-initiation complex for translation tries to start translating, it's not going to be able to plow through these RNA protein structures. So you're inhibiting translation initiation. If If you can't get through the 5' UTR, you're not going to make any protein. So that is how we block ferritin. It's at the translational level when there are low levels of iron. Now let's look at the situation for the transferrin receptor messenger RNA. So here it's the opposite, right? So if we have high levels of iron, we don't want to bring more into the cell. We don't want this receptor to be on. We don't want more iron coming in. So the transferrin receptor messenger RNA actually has its IREs in its 3' UTR. So you can remember transferrin begins with a T, 3' UTR. So it has multiple IREs in its 3' UTR, and it turns out that there is a sequence in the IREs that is AU-rich. What do AU-rich sequences do? Yes, they bring in enzymes that destabilize the messenger RNA. They usually recruit deadenylases. So these are not being bound by the RNA binding protein because it is stuck to iron. So they are visible to those AU-rich binding proteins that are going to come in, cause degradation of the messenger RNA. You're not going to be able to make transferrin receptor. All right, but what about when iron levels go low? Now we have the free RNA binding protein, and it can now bind to those IREs and hide that AU-rich sequence so the destabilizing enzymes no longer see it. These are in the 3' UTR. Translation initiation can occur. You decode, stop codon is here, and now you don't get degradation, and instead you get high levels of this protein being made because the IREBP is bound to the 3' UTR and stabilizing the RNA. But we saw with ferritin, when the IREBP was bound to the messenger RNA, it was blocking translation. Why isn't it blocking translation here? Why, when it's bound to the 3' UTR? Exactly, yeah. The ribosome never even comes through here, right? It is the untranslated region, and it is downstream of the stop codon. Here, we had it in the 5' UTR. The pre-initiation complex needs to scan through this in order for translation to start. So this is where the position of those IREs is important. The other thing you might be wondering is, well, why doesn't this get degraded, right? Shouldn't there be AU-rich sequences in these IREs? It turns out that there is not. So there's a little bit of difference in the sequence of the IREs in the ferritin versus the transferrin receptor. If you look at the IREs, they form pretty much the same shape, these stem loops. Some sequences, if you look in the loop region, they are identical, and this is important for that RNA binding protein to bind. But if you look in this region right here where there's this AU-rich sequence, you don't see that over here in the ferritin one. So the AU-rich binding proteins see the transferrin receptor IREs that are in the 3' UTR, cause degradation. It is different for the ferritin ones that are in the 5' UTR. Any questions about that? Yes, Nick?
So if it's in the 3' UTR region, it's not affecting translation, what is it doing in the 3' UTR?
So we have, for the transferrin receptor mRNA, we have the IREs that are in the 3' UTR. When they are not bound and protected by the RNA binding protein, what happens to this messenger RNA? Yeah, those AU-rich sequences are available for the AU-rich binding proteins to bind and degrade the messenger RNA. So this RNA is being regulated at the level of stability. When you do not have the IREBP bound, you get the AU-rich binding proteins degrade the messenger RNA. If you don't have messenger RNA, you have nothing to make protein for, right? If the IREBP loses iron, iron levels go low, this is now free, it is going to hide that AU-rich sequence by binding to the IREs. These are no longer recognized by the AU-rich proteins that would degrade the RNA, so the RNA is now stable and can be translated. Does that make sense?
Yeah, so it's translated to create more
it is translated to make more transferrin receptor messenger RNA. So the transferrin receptor, it sits in the membrane of cells, pulls iron into the cell. If you have low iron, you want to be pulling iron into the cell. If you have too much iron, you don't want that receptor there anymore. You don't want to pull any more iron into the cell. Make sense? Yeah. All right. All right, let's do a clicker question. So the levels of mRNA and protein. How do we detect messenger RNA? What method do we use? Northern blot. We're also detecting protein here. What method do we use to detect protein? Western. We didn't go through the steps of a Western, and don't worry about that. Just know that a Western is what is used to detect level and size of protein. Very similar to how a Northern blot is used to look at levels and size of an RNA. So here are our results. Here's our Northern blot. Here's our Western blot. And so which of the following possibilities are supported by these data? Well, hopefully. It just says drugs underneath that. And so it's control, 1, 2, 3. Ten more seconds. Final answers. All right, we have a battle between B and E. Everyone always loves more than one of the above, right? All right, let's go through this. Make sure we know how to interpret these based on what we've learned. Who wants to share an answer? Arya? All right, so let's go through A.
So for A, my thought was that since we see that the control RNA is loaded properly, the mRNA wasn't able to be detected. I mean, this could potentially be because it wasn't properly loaded. It also could be for a number of other reasons, like it wasn't able to be detected. But since I saw that the control RNA was properly detected and that the mRNA wasn't, it might be a possibility that the mRNA wasn't properly loaded. So I thought it was A.
Okay, before we go on, does this stand for control RNA or control condition? So we're looking at control condition, meaning no drugs, drug 1, drug 2, drug 3. What should I look for my control RNA? The control RNA here. This is what I'm looking at to make sure I had similar loading, similar purification of the RNA under each one of my conditions. And so maybe I should have called this no drug. Maybe that was like what got confused there. So it's meant to be control condition versus drugs, but if I call this no drug, then I think maybe there would be less confusion. So now, okay. All right, so before we get on to that, so why are you switching your answer then?
Because A is wrong.
Well, why is A wrong?
It's wrong because since we're talking about just the control conditions, the control RNAs are able to be detected. So obviously there just isn't any mRNA in the process. It doesn't mean that it's improperly loaded.
Yeah, so the first thing I always want to look at with my northern as well as in the western in this case is, how does my control look? Do I have evidence that something was loaded in every lane and loaded to the same amount? So this maybe is our ribosomal RNA, which would be highly expressed in every cell. We see similar levels in all of them. Our control protein actually is looking really great too. So when I say the mRNA from cells treated with drug 1 was not properly loaded in the gel, well, I see with drug 1, I'm not detecting any RNA, but I can't really conclude that there was anything improper about the loading or the experiment. So A is not correct. Does everyone understand that now? All right, so moving on. You like B, was it? So why do you like B?
I like B because in drug 2, we're clearly seeing that like, the mRNA is properly there because of the low band. However, protein X isn't present, and mRNA is part of the process that creates like the polypeptides of proteins. So obviously there has to be something that's not causing the protein to be produced. So I'm leading to believe that it's somehow inhibiting the translation of mRNA-X. mRNA-X is present, but the protein.
Yeah, so that's absolutely possible here. So if we have something that inhibited the translation of the mRNA that's present, we would expect to see no protein, and that's what we see there. So B is correct. What about C? C says alternative splicing induced by drug 3 changed the sequence of mRNA, but not the sequence of the protein for gene X. So we look up at drug 3 and we're comparing to the no drug control. Could alternative splicing happen here? Yeah, alternative splicing can change the size of the messenger RNA. You can skip exons, you could do something. So, all right, we could get a different sized RNA here. What about our protein?
The protein, like, there is a band, but it's in a different position than the control is, which is leading to believe that it's a different sequence. However, the answer says put it on the sequence, so it's wrong.
Yeah, if you see a significantly differently sized protein, you have changed what amino acids are included in that protein. And so you have changed also the protein. So seeing that not in there is what made that one incorrect. Does everyone understand C? What about D? D says drug 2 activates a protein that stimulates the degradation of mRNA-X.
mRNA-X is present in drug 2, so it's not being inhibited.
And so what would you change in this question, one word, to make it correct? So it could either activates a protein that stimulates the degradation of the protein instead of the mRNA. That would have been correct with that, exactly. Anyone have any questions about interpreting this? All right, so why don't we pick up with the rest of these few more slides next Thursday, I guess. Have a good holiday. Absolutely.
I don't understand why, A is wrong.
Okay. So I have it up here still.
Because, I know there's a control, but why can't it be that you loaded your control right and you didn't load protein, 1 right?
Or you mean the RNA? Right.
Yeah.
We're talking about the RNA.
Yeah, the RNA,
So
sorry.
do you remember how when we talked about the way that we reprobe a gel for the control? So what you can do is you can do your northern blot. And you saw nothing. Okay, that could be because I didn't load it or because it wasn't expressed. So then what you do is you strip off the probe, you throw the boiling water onto your blot, you get rid of that probe for mRNA-1, and now you throw the probe on for your control RNA.
Oh, so it's
you
the
haven't
same, you haven't done anything to the RNA.
exactly. It's the same exact sample of RNA on the same exact blot. And I realize that